Today, I'm just going to talk about some comparative study on the five matter pathway connecting those and ventral visual cortex. So some of the data are relatively old, and some of the data are just almost close to be published. Well, let's just this is the outline of talk today. First, I'm just make a very brief introduction for maybe biological vision and five matter. Then, secondly, I'm just going to talk about some vertical occipital fasciculus concept and study on the humans. Then later, we are just going to talk about non-human primates. Then in the final part, includes relatively new data, including maybe comparison across mammalian species. So let's start. Well, I think we are getting some later end of some maybe symposium. Some of you are just getting a little tired. So I just want to start something fun. Well, you look slightly tired. Just let's enjoy. So this is kind of phenomenon our postdoc Junshan Lo discovered. Just point out this number, which number you recognize. Speak out. 6. Yes, good. So there's some mixed response between 6 and 8. It is correct because this just occluding mass make it ambiguous. You can actually interpret them both ways. Then this is called some bistable perception, perceptual ambiguity. I can just give you other demo. This is kind of United States, just 3, then you can see signal and counting down. 10, 5, 6, 7, 8, 9. What? I just tried to hack you. And actually, it's everything counting down because signal clock, but because it's just partly hidden, you can just actually interpret in a different way. And then this is kind of phenomenon we discover. We do psychophysics about this, there's a poster about this. But actually, the point here is our brain is just interpreting just incoming signal because incoming signal is usually incomplete. So we have to have some biological system to interpret what's just coming from our retina, from our input, just try to extract meaningful information, which is just important for our just behavior survival. Then which is just important in many ways because the vision is a basis for various cognitive function, including avoiding some danger or maybe try to get the food and also maybe try to make social communication. And also there are many social impact to understand biological visual system because Ariel talked about glaucoma yesterday, but prevalence of the glaucoma in this country is particularly very high because once we get just 70, I think prevalence is just about 10%. So nevertheless, so we are motivated to study about biological vision. Then I just raised 2 major research questions in these talks. First is, what is the appropriate level of the biological complexity to be included in the model of the neural computation, the visual system? Because we have an inherent trade-off between biologically accurate, or it's just more just easy to interpret. Then we just try to reach a level which is kind of meaningful, appropriate to understand biological visual architecture. Then second question is how much we can just incorporate some neuroanatomical knowledge to guide biologically plausible model of the neural computation. Because people usually say vision is the most well studied in the system, in the brain. It's just well investigated, it's well discovered, partly true, partly not. Well, getting back to the 1982, Leslie Younger, and Mishkin just proposed this kind of theory, which is called two-stream hypothesis, which is generally distinguish some dorsal visual cortex, receiving some spatial information and also guiding action. Ventral part of the visual cortex is handling interpretation of the color, form, face, object, some categorical information. So by and large, this kind of category of the dorsal stream and ventral stream are widely accepted among many neuroscientists. So what are these should be reflected in actual model or architecture of our visual process in the brain? So we just open some textbook for undergrad student in grad school. Let's just open Candel books. So we can see the simplest form of the model of the cartoon. Say signal just coming from LGN or B1, it just go to MT and dorsal, then just B1 to B4 to IT cortex. This is it. Then of course we know this is overly simplistic because there are many other area important connections which are missing from our textbook. Or this is the other form, maybe this is kind of motivated from some macaque electrophysiological finding. Other kind of line model emphasize more feed-forward processing from ganglion cell, LGN, B1, B2, B4, IT cortex. Well, this is actually affecting many of kind of current neural inspired convolutional neural network type of model of visual processing, which often emphasize B1, B2, B4, IT cortex. But I'm not sure these are kind of really appropriately biological plausible model for our visual processing behind, because these models ignore existing many anatomical pathway and also existence of some major area, because B3 often ignored and B3A often ignored. But I think the other way to describe visual architecture, this already shown up twice in this symposium, this Federman and Bayesen diagram. So this is a famous, it's a complex diagram of the anatomical connection coming after the collection of the macaque tracer study. So talking about my hierarchical structure, but anatomy is complex. But people usually use this cartoon to talk about how anatomy is complex, but I think it's very essential to build a model of how these are connected. Because how these are connected means I think we need to build some model to understand anatomical wiring. Then Steph talking about some model in the language system at the level of the fiber tract. Then we also need some revisit some current understanding of some anatomical wiring and try to think about how to format a tract as a meaningful unit to basically understand what's a biological architecture to think about visual processing. Because as already repeatedly shown this symposium, because why matter tract matters in many cases, it's of course not functionally homogeneous bundle, but once we want to understand some impact of why matter region or maybe information architecture, I think there are many advantages to think about why matter connection at the unit of the why matter bundle. Yes, then this is just an example. Well, this may be already too much because already just repeated a couple of times, because we know that loss of particular white matter tract causing some disconnection syndrome or particular brain function in the older study. Modern cases, we can just use diffusion MRI to just measure white matter bundle from living human brain. Already previous talk already talked enough about the method. I can just skip detail. This is one example about SF1, 2, 3 from Michel's study. Anyway, so just we are moving to the vertical occipital fasciculus. It's actually showed up in the step slide a couple of times, but getting back to the literature, this is a white matter pathway appeared in the late 19th century book written by neuroanatomists at that time, like Wernicke, Obersteiner, Heinrich Sachs, or Dejerin. Then they're just kind of consistent to appear in the multiple books at that time. That basically in the old books say this is a pathway connecting dorsal and ventral occipital cortex. Then interestingly, while this was already reported in the late 19th century, it was largely ignored until recent resurgent interest coming up from diffusion imaging from maybe 2013. Then, because of that reason, I think maybe for long years, I think this was largely underlooked. Therefore, anatomical details and the interspecies homology of this pathway has been largely unknown. Then I was a postdoc in Brian Vandel lab at Stanford, and at that time people were just getting excited to see this pathway. This is one of the paper from former colleague Jason Yetman. So they're just trying to measure pathway around maybe visual waveform area in the ventral temporal occipital cortex. Then they incidentally find maybe streamline coming from diffusion data connecting dorsal to ventral. Then seemingly the BOF appear in the classic book. So then we just wonder, so we want to better characterize how the human BOF is related with early retinotopic map in occipital cortex to better understand how this pathway matter to us to think about the wiring system in visual area to area. This is already a kind of slightly older study, but for this study, we acquire fMRI-based retinopic map and diffusion MRI data from same individual in human. Then we could, of course, identify using tractography the vertical occipital fasciculus in a very reasonable manner. This picture is a coronal section. Then you can see coronal section. So you can see calcanein sulcus here. Then BOF appears at the dorsal to lateral ventral side connection. Then in this picture, it's kind of sagittal view. Then we can see BOF is lateral from optic radiation. This is geniculocortical pathway. The arcuate fasciculus is just more anterior. Then we can just nicely replicate order finding by Dejerin. Then also showing in the coronal section in their dissection figure is the connection between dorsal and lateral ventral visual cortex. But what we're going to do is we want to analyze some cortical area near endpoint of the BOF. I'm just using some nuanced terminology because we know that using diffusion tractography, we couldn't just track carefully into gray matter all the way. There are some limitation, but I just say there's some cortical area with some proximity of the fiber bundle. So we couldn't just make 100% correct fiber project in gray matter. This is an estimate, but we can just use the border between different visual area using retinopic mapping. We can see reversal of the polar angle mapping. Then we can delineate the border between area on the cortical surface. Then we are now looking at dorsal side of the visual cortex. Then we just project the voxel near the dorsal end of the BOF, just streamline as a color heat map. Then in this paper, we consistently found then across subject, B3AB is an area which is just very near the dorsal end of the vertical occipital fasciculus, then also includes some neighboring map. So let's just turn the brain, then looking at the ventral side from the bottom. Then again, we can delineate border between visual area based on retinotopic sign reversal. And then our observation is consistent across hemisphere to hemisphere. So we can just see fourth visual area, we term HB4, consistently appear near the BOF endpoint. In the ventral side of the surface, then this BOF endpoint is near other neighboring map, but we consistently see HB4. So this has been our observation so far, but luckily a couple of people just try to replicate this finding later on. This is a paper by Xu and colleagues. So they are just using different independent diffusion imaging data together with cytoarchitectonic-based some atlas space to compare cortical voxel near the BOF endpoint. They're just parcellation scheme. As you can see it in their data as well, the dorsal end of the BOF are near B3A and B. Then this is cytoarchitecture. It's just use nomenclature HOC4B. But this is B4 equivalent, basically. Then you can see this area is kind of central to ventral end of the BOF. Stephanie just introduced our Kringler dissection method. Then this is the other paper from Japanese Chiba University group. So I haven't met with them until recently. That's interesting. So, but basically, you cannot measure retinotopy in the post-mortem brain. However, they just try to find out sulcus landmark, which is known to be close to some particular retinopic area. They just decided from where their dissection started. Their dissection can start around B3AB, or they just discover vertical fasciculus. Then in the ventral side, we also know some territory south side named PTCOS, which is known to be a landmark for B4 as a vertical ventral occipital map. You can just visually trace down to near HB4BO1 boundary. So seemingly consistent with my original observation. So getting back to original observation, the observation from our diffusion data and a couple of other groups in human, so we can see some BOF across different group, dataset modality. Then it apparently looks like this. B3, AB, HB4 are kind of central for communication between dorsal and ventral. Of course, there are some other connection. Then we only focus on the BOF, and of course, we should be aware that there should be other short distance U fiber connecting near area each other. Then we can just imagine that information architecture looks like this. But I think people just come to me often say, this is very weird. Maybe this may be wrong because I think it should be B1, B2, B4, IT cortex. Well, I don't know. But there are just many papers in the literature talking about B1, B2, B4 IT cortex literature, but I honestly think that one of the reasons is these are just relatively easy area to measure in electrophysiology in the folded macaque brains. Because B3 in the macaque is very difficult to measure its lunate sulcus. But in order to think about how much some architecture of the BOF is applicable to non-human primates, it is very important to extend to non-human primate study, not just validate BOF finding, but it's just very important to get some deeper insight how this architecture emerges as a biological visual system. Because as I remember, Carl Zeus often told me, then Brodmann really emphasize some comparative study, because just looking at one species, you could make some concrete observation, but comparison across species give you some deeper understanding. So just let's get it to there. So the first thing we did it. So we had a chance to collab with a couple of macaque monkey group. This is the data David Leopold and Frank Ye at NIH just generously provided us. This is ex vivo diffusion data acquired from post-mortem fixed macaque monkey brain tissue with 250 micron box cells. Then I could explain this color coding. This color coding called DexMark, diffusion encoded color map, where color red means the diffusion direction to left to right, green is anterior to posterior, blue is superior to inferior. So that you can see this is axial section, then in medial section you can see lots of green. So where many geniculocortical pathways are coming through to anterior posterior direction, then just lateral to it. So I just highlighted here by yellow color, we can see a group of the box cell with blue color. So indicating there should be fiber bundle going to superior to inferior orientation. Then I'm just cutting the section here broadly, then showing coronal section here. Then you can see some group of blue to purple box cell located in the lateral side of the white matter, lateral adjacent to occipital optic radiation. Then you can see some continuous diffusion signal to dorsal to ventral, occipital ventral visual cortex in macaque monkey brain. So this is something for fun. So if I'm correct, if I just try to trace down history to the older date, I think that Caldwell Nikke made the first observation about this pathway in monkey. Then we couldn't find a clear description of which monkey species precisely. It's some old world monkey. But I can see this camera lucida drawing at that time. It seems to be a beautiful picture. I found this picture is very beautiful. I just try to find out similar slice in the diffusion data as possible as I can. Then you can see this sulcus landmark here, superior temporal sulcus, inferior occipital sulcus. Then he described this area surrounded by just blue. I just highlighted its FP. So this is a BOF. Then you can see just blue color in this kind of triangular area laterally adjacent to some geniculocortical pathway. So I would say this is a very nice replication of our original Well-Nikke finding. But interestingly, just people have kind of ignored the existence of this pathway for a long time as a fiber bundle. So then we just try to use tractography using a method Franco briefly mentioned, so our 2016 paper. But I think the point of tractography is we can just visually see fiber orientation in the two-dimensional DexMark precisely without many assumption. But here we make some algorithmic assumption to reconstruct three-dimensional pathway in a volumetric space. Then we just try to do the same analysis we have done for human. We basically take some monkey brain to construct the surfaces. Then we parcellate or delineate border between different visual areas based on cytoarchitecture-based parcellation proposed by Salim and Rosettes. Then we overlay some area near to B4F. Then we could see some areas, say B3 dorsal, B3A, B4 dorsal, near dorsal BF endpoint, whereas ventral side B4 and TO are near ventral endpoint. Then direct one-to-one comparison with humans is complicated. I can tell you maybe long story because people often disagree about homology about one area in the one species or other species in the visual neuroscience because we don't use some nomenclature TO in the human by many reasons. So it's just complex to compare. But there are some general consensus this just pathway exists. Then this connection between dorsal and ventral appear at maybe some mid-level visual processing, say B3, B4. If we are checking some tracer papers, say Leslie Ungerleider, a couple of others, then the anatomical wiring between B3A and B4 ventral are reported as a bidirectional pathway using just tracer injection. But the fact this pathway is forming some bundle connecting dorsal to ventral was not well characterized because this is just only describing the endpoints after the tracer injection. So here we could combine human just dissection literature, diffusion literature, then coming back to the macaque diffusion data, we just try to get converging evidence from different approach. We also extended our approach to common marmoset brain. Already appeared a couple of times in the symposium. We again collaborated with David Leopold, team, and Afonso Silver, Frank Ye, to analyze some high-resolution ex vivo diffusion data from marmoset brain. So what is interesting about marmoset is their number of the sulcus folding is just very limited. It looks more flat. So because of this simple architecture, then white matter bundle organization in the diffusion data looks very simplistic. Because you can just see green in here, it's blue here, overall very similar because it looks like BOF, but because there's no sulcus folding dividing phi matter, so there's sulcus don't penetrate the phi matter, then this blue area, BOF, looks very continuous sheet from very anterior to posterior, unlike human macaque. So it just appear near B3 anteriorly, then it just continue to parietal to infrafrontal cortex just anteriorly. But that indicates, getting back to some more MOSFET level, we can also see some connection motif connecting some dorsal and ventral side of visual cortex. But I have to note that some of these just connection between different parts of dorsal to ventral side of occipital cortex is reported by Marcello's paper back to 2009, showing some retinotopy specific just anatomical connection between dorsal and ventral in marmoset. So later, I had a chance to collaborate with the Zurich team. It's wonderful, Dr. Chels, Nicola, and Marcus Axel. So we had a chance to look into the data using some polarized light imaging altogether. This is a technique developed by Zurich team. Mostly, it's a measurement of birefringence of myelinated fiber using polarized microscopy. Because we know that myelinated fiber have optical property called birefringence, then we know that this is dependent on orientation of the axon. Therefore, if we are rotating some polarizer during some data acquisition, then each pixel people can typically get sinusoidal signal from there. Then this phase information, the sinusoidal signal, is corresponding with what people call fiber orientation map. So the advantage here is you can go super high resolution in this approach, unlike some diffusion, even push higher than ex vivo diffusion. But I think it's just still, unlike any of the staining approach, data format is just closest to diffusion. You can see colored fiber orientation, but with better representation in terms of spatial resolution. And other advantage compared to maybe other optical approach, maybe slightly shown up in the Franco slide, well, I think the PLI has a very larger field of view. So you don't have to restrict your just measurement the small piece of the brain. You can just measure entire section of some primate brain, for example, or human brains. So this is a picture I just looked into with Nicola, Carl, and Maxer. Then confusing part here is diffusion people and PLI people don't use the same color coordinate system. There are many reasons behind, but this pinwheel color system is HSV coordinate in PLI color coordinate system. Here, I think fiber passing through the plane is the black. The cyan color is the direction of the dorsal to ventral side of the visual cortex. But looking at this vertebrate monkey brain and coronal section, then this is kind of lateral to the optic radiation. We can distinguish a group of cyan pixel, indicating there are kind of direct fiber connecting dorsal to ventral occipital visual cortex. So we just not only confirm the finding in the diffusion data, we can just zoom into near the cortex to check which just cortical area the fibers can be directly penetrating into. This is very difficult in diffusion MRI because the resolution, but here we can just directly see the observation. Then we just find some consistency across the data set about this mid-level area-to-area connected between dorsal and ventral. We also measure sagittary section data and then also confirm this observation. So interim summary here. So the BOF is commonly observed in human macaque marmoset diffusion data. Then it just appears at around B3, B4 posteriorly, and it just continues to further anterior region. But depending on the species, sometimes it just branched between anterior to posterior segment, but marmoset, it looks very continuous. Then it's also very visible in the high resolution anatomical data from vertebrate monkey. Then this finding supports a view like some visual processing architecture found in human BOF is also prominent in non-human primates. Just. So the final question is how much this just organization principle of the vertical occipital fasciculus can be applicable to other mammalian species. Then I think that this paper is just very recently accepted. We collaborate with some Chet Sherwood, maybe Marcus Axel, Erin Hecht, David Leopold, and Frankie and Takaaki Kaneko. So we just compare diffusion data acquired from 12 mammalian species, which are kind of strategically selected along some evolutionary tree to better understand some evolutionary just origin of occipital fimatal tracts. So then this is kind of cartoon about evolutionary tree, but I will just try to begin from primate species here, because we already investigated some macaque monkey and marmoset here. We included some rango and hora monkey. So there are also different type of primate species. Then this color coding is basically the same as I showed in the macaque slide before, because red is left to right, green is anterior posterior, blue is superior to inferior. Then what we can consistently find some organization principle is largely preserved here, because medial optic radiation green, then we can see some group of some blue voxel connecting dorsal to ventral most lateral side. Then medial to optic radiation, we see some tapetum, and then which is some continuum of the callosal fiber coming from occipital corpus callosum, then coming near calcareous sulcus. This architecture like tapetum in the medial, the next level is geniculocortical pathway, then most laterally we see the voxel from with dorsal to ventral diffusion. So this is some principle we commonly seen in the many of some diffusion data in the primate species. So suggesting some preservation. So we push just one way further to looking at the two species, Strepsirrhini species, which are still within primate trees, but which are more distant from human compared to macaque monkey and marmoset. Then these are kind of data we could just get from NIH scanner. Then top one is Remo, then bottom one is Garago, then here's the axial section. Here you can see coronal section. But again, I think it looks largely similar because we can see some geniculocortical pathway green here. Most medial side is kind of tapetum, forming some maybe callosal fiber here. Then most lateral side, we can see some blue to purple voxel surrounding optic radiation, then which looks kind of similar in the Garago, which is kind of very far from human, but within still primates. Then we can see group of blue to purple voxel traveling superior to inferior direction in the lateral side of the white matter in the occipital cortex. So the question is, if we are looking at non-primates, how much this white matter architecture or organization principle with the spatial layout can be preserved? So for this reason, we just looked into four non-primate species. These are tree shrew, ground squirrel, rat, rodent, paca. Then I just pick three pictures here. I just get used to the human brain too much. Then sorry about it, but looking at some rodent brain, I just get lost to some extent because there are some lots of rotational difference because visual cortex on the top, hippocampus going on the top, many things are different. But we just took a simplest strategy. We just place the seed in the primary visual cortex, dorsal lateral genicular nucleus, where I can confidently observe. I just try to identify pathway connecting them as an optic radiation equivalent here to mark voxel. Then we just try to discover voxel perpendicularly and laterally adjacent to those optic radiation. Then we conclude that it's so difficult to find that lateral compartment. Because you see some in rat data from Alan Johnston in Duke, they have very magnified callosal fiber. Then large portion of the callosal fiber is there. We can see some geniculocortical portion, some medial callosal, some geniculocortical, but it's very difficult to find third component appear lateral to optic radiation, travel to superior to inferior axis. So finally, we also look into two carnivorous species, which is kind of slightly far, even far from the rats, but they have some architecture similar to the primate in a sense of more folding. We know that ferret visual system have some preservation of the visual function, like fovea-like structure in the retina. So we also looked into ferret and fox. Again, we look into some diffusion color maps here. So our observation here is we actually do not find white matter tract as prominent as primate BOF in these data set. But interestingly, we see some small voxel here looking at purple and blue here, seemingly kind of BOF, which is the ferret, which is kind of slightly confusing. But our observation is this kind of colored area connecting dorsal to ventral orientation seems to fall within more gray matter territory. So it's possible that maybe similar connection may be prevalent, but it just follows as a horizontal connection in the gray matter. That's a speculation. But overall, in Fox data, we also don't see clear sign of the BOF in there. This suggests that even if maybe those pathways present, they are less prominent compared to that in primates. So I just need to conclude because it's slightly tricky to interpret null finding. How to interpret apparent lack of the evidence that BOF in non-primates? Of course, logically, we can't say it doesn't exist. It's just logically impossible. But one possibility is maybe similar track may be preserved in non-primates, but it is too small to identify. So to address this question, we also collaborate with Marcus Axel. So they have high resolution PRI data already acquired from rat brain. So then he provided some coronary section rat data as well as kind of sagittary section male rat PRI data acquired from some different brain. Then you see some overall some deck mapping diffusion data, fiber orientation map PRI has some good agreement each other. But again, it's very difficult to find out some prominent pathway traveling dorsal to ventral in some rat data. It's kind of very difficult to find out. Still, we can't say it doesn't exist. But this is kind of 1.5 micrometer pixel resolution, even pushing the limit. So we couldn't reach the level to identify similar pathway as much we can clearly see in many of the primate data in much coarse resolution. So that's one reason I can still kick out this. But we are starting to think about it's kind of relatively less likely. So then I will actually finish my talk by raising 3 other potential possibilities. Then first possibility, this is quite likely. A homologous axonal pathway in non-primates exists, but they are running through the gray matter rather than white matter. I think this is possible because there are kind of recent brain structure paper from Watakabe, this Japanese anatomist. They say if we are looking at some rodent neuroanatomy data, I think majority of the carousel pathway, I think across hemisphere pathway, they are coming out to the white matter. But if we are looking into within hemisphere connection, majority of the axons stay in the gray matter. So this is their observation. Then they say this principle is different from those in the primates. From this principle, it's likely that this kind of visual to video connection staying in the gray matter more in the rodent brain. Or maybe other possibility is this. So similar pathway exists, then it's coming out of white matter, but they have fundamentally different orientation from primates. Because compared to primates and non-primates, we see some overall rotation of functionally homological area. Because human and macaque, mammoth, we have all some backside of the brain and visual cortex. Then rodent, I think it's just coming out to the top. So we people typically see some rotational relationships, some homologous structure location in terms of geometry is not fully consistent across maybe rodent and primates. So therefore, if we see some similar pathway in the rodent, I think their orientation can be different. But if the orientation is similar to other pathways that are running parallel, we have no chance to identify using existing methodology to check some fiber orientation. This is the one possibility. Then I'm not going to strongly push the last possibility, but of course it's interesting to think about this pathway may be relevant for primate specialization of the visual function, but this is the one with the many possibilities to interpret. Because if we are thinking about what is special about primate visual function, one major sophisticated of the primate species have some visually guided grasping. Because as you can see, human can just grasp just object easily by just looking at by eye. The same for marmoset behavior. Then this activity require information exchange guiding action as well as involving object recognition. But this is partly what makes us human, because we can just visually guide and getting an object. This is not very typical in the rodent because they are just using whisker more to navigate the world to just try to reach. So this is just a hypothesis. It's not proven, but one idea can be evolution. The pathway may be related to some primate specialization in the visually guided behavior, because which require more information exchange between so-called dorsal pathway and ventral pathway. So I'm running short of time to wrap up. So the detailed analysis of occipital white matter suggests there is a prominent fiber pathway connecting dorsal and ventral visual cortex, we term BOF from classical term. Then prominence of this pathway in primates might suggest potential role in the visual guided behavior, which need to be further tested in the future. Then incorporating this pathway into the computational model of the visual processing, I think this is an important next target for visual neuroscience, because we don't want to be too complicated, but we need to just go beyond overly simplistic model, which just ignore many important visual area and pathway. So I'd like to thank all lab members and maybe all international collaborators. They are all wonderful, and thank you very much. And I'd like happy to take any questions.